Welcome back. Chapter number 2, Sexual Reproduction in Flowering Plants. One mark questions. How is bagging technique useful in plant breeding program? Bagging prevents contamination of stigma with unwanted pollen. What is the function of tapetum? Tapetum nourishes the developing pollen grain. What is polyembryony? Poly means many. Embryony means formation of embryo. So, the presence of more than one embryo in a seed is called polyembryony. What is emasculation? The process of removal of anthers from the flower bud is called emasculation. Wind pollinated flowers have to produce enormous amount of pollen. Why? Because they have to be transported in wind current. What is apomixis? Some plants produce seeds without fertilization in their flowers. It is called apomixis. This is one important question. In many question paper, this question has been asked. Name the innermost wall layer of microsporangium, that is tapetum. What are tassels? This is also one important question. Tassels are stigma and style which wave in the wind to trap the pollen grain. Tassels are stigma and style which wave in the wind to trap the pollen grain. So this is important. What are filiform apparatus? Filiform apparatus are present at the micropylar end of the synergids and guides the entry of pollen tube into embryo sac. So that is a function. It will guide the entry of pollen tube into the embryo sac. Give an example for free nuclear endosperm, that is coconut water from the tender coconut. What is meant by monosporic development of female gametophyte? So mono means single. Mono means single. The process of embryo sac formation from a single megaspore is called monosporic development. That is formation of embryo sac from a single megaspore. One single megaspore will lead to the formation of the embryo sac. So that is called as monosporic development. What is heterostyle? Hetero means different, many. So hetero, it is a outbreeding device. So this word is important. I repeat, it is a outbreeding device in which the anther and stigma are placed at different position so that the pollen cannot come in contact with the stigma of the same flower. So here it is one outbreeding device in order to reduce the self-fertilization or to prevent the self-fertilization. Suppose the stamen is below. Suppose this is one stamen in the flower and the stigma is above. Heterostyle, that is at the different position. Suppose this is a stigma and this is a stamen. So stamen cannot go from down to above, means against gravity it won't travel. So that is why, so this is one condition in which the self-fertilization is prevented. Name the asexual reproduction that mimics a sexual reproduction, that is apomixis. This is one important term you have to remember. It will mimic the sexual reproduction. Even though it is a asexual reproduction, it will mimic the sexual reproduction. Name the tallest flower, that is Amorphophallus. Name the homogeneous tissue present in the young anther, that is sporogenous tissue. Define microsporogenesis. So again, this is one important question. The process of formation of microspore from pollen mother cell through meiosis is called microsporogenesis. Name the male gametophyte of angiosperm, that is pollen grain. Mention the chemical composition of exine, that is Sporopollenin. So this is also an important word you have to remember. Sporopollenin. Name the component of pollen grain that forms pollen tube. That is vegetative cell. Define megasporogenesis. The process of formation of megaspore from megaspore mother cell is called megasporogenesis. Name the basal part of ovule, that is chalaza. Name the triploid cell formed due to double fertilization. Primary endosperm nucleus, or it is abbreviated as PEN. Primary endosperm nucleus. The next second main 2-mark question. Name the 4 wall layers of microsporangium, that is outer epidermis. Inner to epidermis there is endothecium, the middle layer and the innermost tapetum. Mention the strategies evolved by flowering plants to attract insects for pollination. Majority of insect pollinated flowers are large, colorful, fragrant and rich in nectar. Mention one harmful and useful effects of pollen. Pollen grains of many species cause severe allergies and respiratory disorders. So that is harmful. Pollen grains are rich in nutrients and is used as food supplement. That is one useful effect of pollen. What is PEN? Mention its function. Primary endosperm nucleus is a triploid cell formed by the union of male gamete with polar nuclei. It forms the endosperm and nourishes the growing embryo. Mention the sequential stages of embryogeny in angiosperm. So this is one important question. You can expect this question in kcet as well as the NEET question. Sequence of embryogeny. That is, first is proembryo, globular embryo, heart-shaped embryo and then the mature embryo. So this you have to write in the sequence. First is pro, then globular, then heart-shaped and then the mature embryo. Next question. Differentiate perisperm and pericarp. In some seeds like black pepper, remnants of nucellus is present. Such residual persistent nucellus is called as the perisperm. The wall of the ovary develops into the wall of fruit and this fruit wall you call it as a pericarp. Differentiate between albuminous and non-albuminous seeds with example. Albuminous seeds retain a part of endosperm as it is not completely used up during the embryo development. For example, castor. Non-albuminous do not have any endosperm as it is completely used up during the development of the embryo. So, for example, pea. Then what are parthenocarpic fruits? Give example. The fruits developed without fertilization are called parthenocarpic fruits. For example, banana. Name the 4 types of cells found in embryo sac of angiosperms. Antipodals, polar nuclei, egg cell and synergids. Write a note on the pollination mechanism in case of Vallisneria. In Vallisneria, the female flower reaches the surface of water by the long stalk and the male flowers or the pollen grains are released onto the surface of water. They are carried passively by water currents. That is, there is no need of any extra energy and some of them eventually reach the female flowers and the stigma. So this is how the pollination mechanism happens in case of the Vallisneria. So this is also one important question. So see the question whether they have asked with the diagram or they have just asked you the explanation. Differentiate between hypocotyle and epicotyle. The cylindrical portion below the level of cotyledon is hypocotyle. The one below the cylindrical portion of this is a cotyledon, below that you call it as a hypo, and that terminates at its lower end in the radicle or root tip. Lower end. The portion of embryonal axis above the level of cotyledon. Suppose if this is a cotyledon, above that it is called as a epicotyle, which terminates with the plumule or the stem tip. So you know that plumule will give rise to shoots and the radicle will give rise to roots. The one roots which are below, that is hypocotyle. Cotyle means below the cotyledon. Below the cotyledon you call it as a hypocotyle, and above the cotyledon region you call it as a epicotyle. Next, third main 3-mark question. What is artificial hybridization, by which technique it is achieved? Artificial hybridization is a crossing of plants in which it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from contamination by unwanted pollen. This is achieved by emasculation and bagging technique. So this is also an important question. So how this is done? Here the plants are crossed in which it's important to make sure that only the desired pollen grains are used for the pollination and the stigma is protected from the contamination of the unwanted pollen. So for this is achieved by the emasculation and bagging technique. Write a note on the structure of pollen grain. Pollen grain has a prominent two-layered wall. The harder outer layer you call it as a exine. Suppose if this is a pollen grain, so the outer wall layer you call it as a exine and made up of a sporopollenin, which is one of the most resistant organic material known. It has an aperture known as a germ pore. So somewhere you have openings like this. So that is called as a germ pore, where sporopollenin is absent. The inner wall layer is called as a intine. The inner to the exine you call it as a intine, made up of the cellulose and pectin. So this composition is important. The intine is made up of cellulose and pectin, whereas the exine is made up of the sporopollenin, and the part of the exine where the sporopollenin is absent you call it as a germ pore. The cytoplasm of pollen grain contains the vegetative cell and generative cell. The vegetative cell is bigger and helps in the formation of pollen tube. The generative cell is smaller and which divides into 2 male gametes. So this line is very important. The generative cell is smaller and it will form the 2 male gametes, whereas the vegetative cell is slightly bigger and it will form the pollen tube. So this is very important for your MCQs as well as the fill up the blanks. Okay. Next, explain the triple fusion and double fertilization in angiosperm. After entering one of the synergids, the pollen tube releases the 2 male gametes into the cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuses with its nucleus, thus completing the syngamy. Syngamy means here it is a fertilization. This results in the formation of a diploid cell called as a zygote. The other male gamete moves towards the 2 polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus. As this involves the fusion of 3 haploid nuclei, it is called triple fusion. So here what are the two polar nuclei? N plus 1 male gamete, it will form a 3N. Whereas in case of the syngamy, 1 male gamete will fuse with the female gamete, it will form a zygote. So that is a difference, syngamy, and then here it is a triple fusion, because here fusion of 3 haploid nuclei is taking place, that is 2 polar nuclei in the central cell and 1 with the fuses with the male gamete. So it will form a triploid, that is a primary endosperm, that is PEN. Since 2 types of fusions, that is syngamy and triple fusion, takes place in the embryo sac, the phenomenon is called as the double fertilization. So this is very important question, students. What is emasculation? When and why does a plant breeder employ this technique? If the female parent plants bears bisexual flowers, removal of the anther is called emasculation. Emasculation is done in the bud condition. So this you have to remember. Once the bud is open, you cannot do the emasculation. Emasculation has to be performed in the bud condition before the bud is open, to make sure that only the desired pollen grains pollinated and the stigma is protected from the unwanted pollen. Next, with reference to monocot seed, define the following. That is, first one they have asked scutellum, alluron layer, epicotyle. So these are important for definitions as well as the 1 mark questions and then the fill up the blanks. The single large cotyledon of monocot seed is called as the scutellum. The covering of the endosperm separating the embryo by a proteinaceous layer in monocot seed is called alluron layer. The portion of the embryonal axis above the level of attachment of scutellum is called as the epicotyle. What is apomixis and what is its importance? Few flowering plants such as some species of Asteraceae and grasses have evolved a special mechanism to produce seeds without fertilization called as a apomixis. If hybrid plants are made to produce seeds by apomixis, the farmers need not purchase the seeds for every season, and the progeny plants will not segregate and will be as their parent. So that is one important. This is also one important question, students. Define autogamy. Write 2 different kinds of flowers that exhibit autogamy. Autogamy is the process of transfer of pollen grain from the anther to the stigma of the same flower. Auto means self. Cleistogamous flowers and chasmogamous flowers are 2 kinds. Which are the 2 kinds of flowers produced during autogamy? That is cleistogamous and the chasmogamous. Write the characteristic features of insect pollinated flowers. So this is important question. Majority of insect pollinated flowers are large, colorful, fragrant and rich in nectar. When the flowers are small, a number of flowers are clustered into an inflorescence to make them conspicuous. The pollen grains are sticky and get adhered to the insects and get carried to another flower. So these are the important characteristic features of the insect pollinated flowers. Draw neat label diagram of TS of young anther. You can see the outer epidermis. Inner to the epidermis you can see the endothecium, and then you can see the 2 middle layers, and you can see the microspore mother cell, tapetum. So below the figure you have to write TS of young anther and then you underline. Then draw neat label diagram of monocot embryo. So you can see the scutellum, coleoptile, shoot apex, epiblast, radicle, root cap, coleorhiza. So this is a diagram of the monocot embryo. So you have to write the footnote and you have to draw the diagram neatly and label it. Unlabeled diagram does not fetch you any marks. Keep in mind, you have to draw the diagram neatly and then you have to label the diagram. Okay. Write 3 advantages offered by the seeds to angiosperms. Since reproductive process such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategies for dispersal to new habitats and help the species own. Hard seed coat provides protection to young embryo. Being products of sexual reproduction, they generate new genetic combinations leading to variation. You can write any 3 points. 4 points I have given you. You can write any 3 points which is easy for you. Answer the following. Compare geitonogamy and xenogamy, which can be asked for 2 mark. What are chasmogamous flower? 1 mark. So both together it is 3 mark. Okay. Geitonogamy is transfer of pollen grains from the anther to the stigma of another flower of the same plant. Suppose if this is one plant, so here one flower is there and here one flower is there. So suppose the anther from this flower is transferred to this flower, stigma. So that means the transfer is taking place within the same plant. Xenogamy is a transfer of pollen grain from anther to stigma of the different plants. So suppose here one plant A will be there, plant B will be there. From here the anther will be transferred to the B plant. Okay. So that is a xenogamy or the cross pollination. Next, second question of the same question, that is chasmogamous flowers are similar to flowers of other species with exposed anthers and stigma. They are seen in Viola, Oxalis, Commelina. So this is important, chasmogamous flowers examples. Next, fourth main, that is 5 mark questions. Explain the structure of megasporangium or describe the structure of anatropous ovule with the help of a neat label diagram. Okay. So this is an anatropous ovule. You have to write the funicle, should show the hilum, micropyle are pole, then outer integument, inner integument. This is a nucellus. And then you have a embryo sac. This is a chalazal pole. Okay. So this is a structure of anatropous ovule. The ovule is a small structure attached to the placenta by means of a stalk called as a funicle. So this is a stalk-like structure, isn't it? So that is funicle. The body of the ovule fuses with the funicle in the region called as a hilum. So in this hilum, it will the funicle will be fusing here with the body of the ovule. Each ovule has 1 or 2 protective envelopes called as integuments. You have the outer integument and then the inner integument. Integuments encircle the ovule except at the tip where a small opening called the micropyle. So here you can see the small opening. This is called as a micropyle. The opening is called as a micropyle. Opposite the micropylar end is the chalaza. So here the opposite to the micropyle. So here it is a micropylar end. So here you can see the chalazal pole representing the basal part of the ovule. Enclosed within the integument is a mass of cells called as the nucellus. So you can see the nucellus here. Mass of cells called as the nucellus. Located in the nucellus is the embryo sac or the female gametophyte. So this is embryo sac or the female gametophyte. Explain outbreeding devices that prevent self-pollination. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. First device is, in some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive or stigma becomes receptive much before the release of the pollen. So this is the first thing. Then the second device is, in some species the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. This condition is called as heterostyle. The third device is to prevent the interbreeding is self-incompatibility. This is one important, self-incompatibility or self-sterility. This is the genetic mechanism and prevents self-pollen from fertilizing the ovules by inhibiting the pollen germination or pollen tube growth in the pistil. Fourth device is to produce unisexual flowers. So the first is synchronization does not happen, the stigma receptivity and the pollen release will not synchronize. The second is the heterostyle, that is they are placed at the different positions so that there is no chance of getting fertilized. The third is the genetic mechanism called as the self-incompatibility, and the last is the production of the unisexual flower. This is highly important question. Please don't leave this question, students. Draw and describe the structure of a matured embryo sac of angiosperm. The embryo sac is present in the ovule surrounded between the nucellus. The embryo sac is 8 nucleate and 7 celled. So this is a mature embryo sac. Okay, so you have 3 cells are at the chalazal end. You can see the chalazal end. There are 3 cells, that is antipodals are 3 in number and are called antipodals. The large central cell has 2 polar nuclei. You can see this is a large central cell. Then at the micropolar end has an egg apparatus, that is egg cells and 2 synergids. You can see the egg cells 1, synergids 2, central cell 1. So 3 plus 1 plus 1 plus 2, that is 3, 4, 5, 6, 7, that is 7 cells and 8 nucleus. You know that in the central cell there is a 2 polar nucleus. So therefore the number of nucleus is 8. 8 nucleate. Next question. What is megasporogenesis? Explain the development of 8 nucleate embryo sac in flowering plants. So the process of formation of megaspores from the megaspore mother cells is called megasporogenesis. Ovules generally differentiate a single megaspore mother cells, that is MMC, in the micropolar region of the nucellus. So you can see the micropolar end, 1 nucellus, then megaspore mother cells. You can see there is a division of the cells, then there is a formation of the megaspore tetrad, that is 1, 2, 3, 4. 4 cells are there, that is why it is called as a tetrad. It is a large cell containing dense cytoplasm and prominent nucleus. The MMC undergoes meiotic division. Meiosis results in the production of 4 megaspores. One of the megaspore is functional while other 3 degenerate. So this you have to remember, out of the 4 megaspores, 1 only is functional and another 3 will degenerate. Functional megaspore develops into female gametophyte. This method of embryo sac formation from a single megaspore is called as monosporic development. You can see the 1 megaspore is forming the formation of the embryo sac. The nucleus of the functional megaspore divides mitotically to form 2 nuclei which move to opposite poles, forming the 2 nucleate embryo sac. The 2 more sequential mitotic nuclear division results in the formation of the 4 nucleate and later the 8 nucleate stages of the embryo sac. You can see the 2 nucleate stage, 4 nucleate stage, 8 nucleate stage. Then you can see the differentiation of the cells. You can see there is a formation of at the micropolar end you can form the egg apparatus, and at the chalazal pole you can see the formation of 3 antipodals.